Hello and welcome to the Xapiens podcast. I'm your host, Loyd Waites. Today I want to start by putting a couple things in perspective. During the Cold War at a U.S. Army base, they were producing vats of anthrax. The idea was to have, instead of a missile that would explode on impact, it would explode producing large amounts of toxins or biological weapons that were capable of destroying an entire population. Now, why was the government doing this? Well, rather than producing nuclear weapons that cost millions or sometimes billions of dollars, it's a lot cheaper to make biological agents to do this and cause just as much destruction. But these weapons never happened. Why? Well, that's in large part due to the guest that I have on today. His name is Mathew Meselon. He's the man who led the International Bioweapons Treaty that prevented these weapons for mass destruction ever from coming to fruition. There have been incidents of biological terrorism in the past, but because of this treaty, because of the work done by Professor Meselson, weapons of mass destruction using biological agents and toxins have never come to bear. Dr. Meselson's CV is insane, and it's amazing to read. When he was in graduate school, he worked with Franklin Stahl to understand and experimentally verify the way that DNA replicates in collaboration with Jim Watson from Watson and Crick fame. Not only that, but he went on to be one of the discoverers of messenger RNA, which you might recognize from being a major part of the COVID vaccine. Today, he's working on projects ranging from dealing with COVID to longevity. And this was a really inspiring and sometimes emotional conversation that I had with him today. So I hope you appreciate it. It was a really moving and interesting conversation that I had with him. Thanks for coming on. You've had a crazy career with a whole bunch of different talking points. So I guess where I can start is, what are you working on now? Do you have current projects?
Yes, several.
Okay.
So we're trying to deal with this pandemic partly by isolation, partly by masks. We used to be told to wash our hands, soaping them for at least 20 seconds. You don't hear that anymore. We use vaccines. They wear off pretty quickly, the RNA vaccines. You have to get boosters, and then variants appear, and they are, in some cases, not very susceptible to the current vaccine. You have to make a new vaccine. A lot of people won't get vaccinated because they don't want to, and that creates a lot of political strife. There are countries where there's just not enough wealth to vaccinate a large fraction of the population. So the virus keeps multiplying in those unvaccinated populations. Not that it doesn't also do so even in vaccinated populations, but especially in unvaccinated ones. And so that's how variants appear. That's why we keep seeing these new variants from South Africa. It's very expensive, and the money goes to parts of our economy that arguably you could say don't really need it. That is the pharmaceutical industry. It's not a very satisfactory situation. We've adapted to it partly just by saying, okay, we'll live with it. It's killed 1 million Americans. I suppose most of those deaths are accompanied by a lot of grief. These are people, loved ones. It's a terrible situation. And yet, this is the only approach that the government or other governments or the WHO or the CDC has envisaged. Now, we used to have various epidemics. For example, cholera. It used to be quite common in England. And it was thought that it was caused by bad air coming off the Thames. till a physician named John Snow took personal histories from patients who had come down with cholera. And what do you know? They were all purchasing water, because you had to buy water then, from the same pump. Couldn't be explained by bad air from the Thames. Had to be waterborne. Well, we clean our water, or at least we sterilize it, with chlorine or with oxygen. I don't think there's a community in America that gets water that isn't in some way or other protected against things like cholera, usually by chlorination. I'm not sure that's right. Maybe these days it's by oxygenation, either one. Why aren't we thinking about preventing the transmission in the first place? How would you do that? Well, you'd have to, first of all, get a bunch of people together to think about it. People who know what they're talking about. And what they would ask themselves is, if we installed filters, and everybody knows about HEPA filters these days, they take out particles 1 micron in diameter and even smaller, which is the size of the droplets of concern. I'll come to that. So we could put them in all the pubs, because at least popular pubs, people get pretty close to each other in a pub, elbow to elbow. Movie houses, some churches, schoolrooms, big apartment complexes, big office buildings. Can we, if we do that, get the reproduction number below 1? That means that if I get infected, on average, the number of people I infect is less than 1. That's on average. Now, I might infect somebody, but on average, only 1. So if you have a number that's less than 1, but still finite, you can have local outbreaks. I might infect somebody, even though on the average, guys like me infect less than 1. But what a great thing it would be if the pandemic was not a pandemic, but instead occasional little outbreaks. Could never threaten a whole economy, could never threaten a whole country. Why aren't we doing this? Well, until just about recently, both the WHO and the CDC argued actively that it is not aerosols. Then they took refuge behind saying, well, maybe it's airborne, but what they were thinking of is what's called droplets. And the terminology here confuses the whole issue. But droplets are the things that you produce when you cough or shout or sing, and they're all produced in the upper respiratory tract. They're not the ones that are infective, and for a good reason, which I'll come to. Do we produce any other particle? Yes, everybody in the course of normal breathing produces much smaller particles. Now, it's not the size that's so important. It's the Stokes law settling velocity. If a particle is small enough, settling under gravity is negligible compared to its ups and downs and backs and forth because of air turbulence. So it's like cigarette smoke. It stays suspended, but they diffuse because it's not Einsteinian diffusion. It's diffusion because the structure of the atmosphere is somewhat turbulent. But mathematically, it's the same thing. Isotropic motions, none of which remembers the That's not quite true. There are some correlations between turbulent flow in the atmosphere, but very crudely, you could say when a particle goes that way, it doesn't remember it did, and the next motion could be any way. And that's real diffusion. Anyway, when we breathe in and out, there are miles of little tiny tubes that lead from the upper respiratory tract down to the alveoli where the gas exchange happens. Carbon dioxide out, oxygen in. And every time you inhale or exhale, these tubes open and close. and they're coated with mucus. So when they open, little strings of mucus are pulled out. And as they continue to open, the strings break. And if you have a string of mucus and you pull it till it breaks, you make one spherical particle. And depending on the surface tension of the mucus, that determines the diameter of the particle, how much work is required to make it. That's where the little particles come from. Those are the particles that can travel all over the place. In a enclosed room like this room, they just stay airborne for a long time. They do deposit on walls. If they pick up an electron, because there are electrons in air, then they probably, if the wall here is positive compared to the particle.
Right, it's static, it'll stick on.
And that's why when you have a picture of your maiden aunt or somebody on the wall for years and years in your house, and you take it down, you find it's all black around the edges on the wall. That's because electrostatic deposition of all kinds of dirty little particles and stuff like that. So that would not only prevent a pandemic in the first place, that is a pandemic that travels by aerosols.
I'm sorry, what was the process that you were saying specifically to prevent the pandemic? What was the...
Filter the air.
Air filters, okay.
Where people congregate. Now what you need to do is to find out how many places, which particular places would need to have filtered air before the propagation coefficient is below 1. Well, different viruses have different propagation coefficients. And not only that, it depends on the temperature of the air and the humidity of the air and pollutants in the air. But anyway, there's a number. So we need to know if it's practicable or if instead you'd have to filter even hermits, even places where single hermits live, But I'm pretty confident, because I've done back-of-the-envelope calculations, that you don't need to filter anything except the places where large numbers of people congregate. And then the other number you need to know is how much it's going to cost. So I should have done this 2 years ago. What you need is somebody who has what you could call convening power, who could bring together 6, 8, 10 people who really know what they're talking about and who are regarded as credible, maybe even authoritative. They write a white paper with saying whether you can do it and what it would cost, and they publish it, and they have a press conference, and then they try and get the White House to do it. I should have done this 2 years ago. I've been saying this for more than 2 years. I sent a letter about this to the New England Journal of Medicine, where they published it. I wanted to publish a whole article, but they were hostile to the idea. The whole medical profession, not just in the United States, but as I said, also represented by the WHO, but worldwide, for reasons that could be explained only by a sociologist, but not by me, have a deep dislike for the idea of airborne infection. By airborne, I mean aerosol-borne. It is generally understood and recognized that measles can be transmitted this way, and TB. But for some reason, there's fierce disbelief. Very sad. But that's another subject. There used to be housed here something called, no longer at MIT, called the Dibner Foundation. Have you ever heard of it?
I have, yeah.
While it was housed here, and my wife was one of the Dibner scholars, she shared an office with a man whose name I don't remember, but his work at the Dibner was to write a book about a man named Wells, who worked at Harvard and then he moved maybe to Notre Dame. He was an early prophet of aerosol-borne infection, but he was treated as a pariah. So I'll leave aside this question because it's outside my domain the sociology of why most physicians really dislike the idea of aerosol-borne infection, and explains why the WHO and the CDC both used to say that it doesn't happen, and now they cloud it by saying airborne, not aerosol, airborne. And that includes two kinds of particles. Those that are produced from big particles, which then evaporate and become smaller. Those are called droplet nuclei. They are the nuclei of what used to be a bigger droplet. That's not what we're talking about. Those come from the upper respiratory tract. What I'm talking about are the ones that come from the opening and closing. And there may be other ways in which these very small particles are produced, but they're produced deep in the lungs. That's important because it works the other direction as well. When you inhale a very small particle, it doesn't have enough mass to have enough kinetic energy to break out of the flow lines of the air you're inhaling and crash into a wall. Instead, it goes all the way down to your alveoli and settles under gravity. Now, those cells in the alveoli, for a reason I don't understand, I don't know if anyone does, have the ACE receptors to which the COVID virus binds. Why do those cells have that receptor? I don't know. But that's how this virus gets into your body, to those receptors. Receptor being a complicated protein structure to which the virus binds and gets injected into the cell. So that's how I believe the infections mainly happen. Not from big particles on your hands that you then touch your nose, not even from big snotty particles that have not yet reached the ground but someone who's coughed or shouted, but from these teeny particles that do not land anywhere until they get all the way down to the alveoli. We need to understand more about this, that's clear. I know something about this because of our biological warfare program. Because there was a time when I shouldn't say we, because it was only a very small and contained part of our government, but their mission was to develop weapons that could infect everybody in Moscow, for example, with something that would kill them. That was the idea. And we did a lot of work on that. And to do that, you had to understand aerosols. And we'll talk about this later, but when I got interested in this, I thought I better learn as much as I can, because when you're debating someone, especially in public, and this is not a very honest trick, it's somewhat underhanded, but if you can show to the listeners, to the audience, that you know more about the subject than the person who's arguing against you, even though their argument might be better than your argument, you've weakened their argument by showing there's something they didn't know that you know. So I quite deliberately decided I'm going to learn more about this than the people who are doing it, which was possible because the people who are doing it usually had a specialty, and I could read across, and I had access to the classified literature, so I could read everything. And I knew enough physics and chemistry to be able to understand what I read. So I know a lot about this. And then when I got interested in this epidemic of anthrax in the Soviet Union, that involves aerosol spread because we didn't know at first how this happened, but it was a release of an aerosol from a military facility that traveled downwind and infected and killed at least 70 people. We didn't know that at first. That's a long story. Anyway, so finally, and I won't go into details because it's still at a very early stage, but I found someone with what I could call convening power, very strong convening power, and this person has started in motion the group of people to write the white paper, to publish in the Proceedings of the National Academy, to have a press conference, to try and to get the White House to say, yeah, we're going to do it. It should have been done 2 years ago. It's very important. Right. The next pandemic there have been several really big pandemics, you know. There's, of course, the the Black Death. There's going farther back, there's the Justinian plague. Then there's a plague of Athens, probably Athens wouldn't have lost out to Sparta if it hadn't been so weakened by that plague, which Thucydides himself, he was infected with it. And he was a very smart man. He asked, mainly he's writing the history of the Peloponnesian Wars, but he asks himself, like a modern epidemiologist would, where did this infection come from? Infection which he himself had. And he knew that it started down, not in Athens, but in the port city, the Piraeus. So that's down south of Athens. So it must have come by sea, he concludes. So he inquires to find out, what kind of traffic was coming in? Well, the Spartans had blockaded ships coming in from the north because they supplied grain from Scythia to the people in Athens. So that it was blockaded by the Spartan navy. So what are these ships bringing in? Mainly, according to Thucydides, tropical birds for the oligarchs. Well, there was a great professor at the Harvard Medical School named John Enders. He got the Nobel Prize for perfecting a system to grow human cells in tissue culture. And he once gave us a lecture on what was called the Marburg virus. This was back in I'm not even sure whether it was the 40s. I guess it was the 60s, maybe the late 50s or 60s. Laboratories in Marburg in Germany, in some place in Yugoslavia, that was before Yugoslavia split, and in a laboratory in England, workers all came down with this viral disease. It killed some of them. So since it occurred in 3 separate places, the obvious question, what is the link? Well, they had all received monkeys from the same monkey farm in Africa. So naturally, you want to go and see if those monkeys on the farm have this virus. They did not. Well, John Enders figured out that since the monkeys were the only common thing in the 3 labs, separated at great distances, Yugoslavia to England, big distance, it must have been something that infected the monkeys after they left Africa. Well, if you go to England with your dog, it has to be quarantined. That's not true here, but the British, you have to quarantine the animals. So John Enders somehow found out they have a big shed where they quarantine all the animals at Heathrow. He found out that the monkeys that came from this farm in Africa and which were put in cages in this big shed at Heathrow Airport, right next to them were cages full of tropical birds. Is it possible that the vector in both the plague of Athens and the Marburg, that the vector was some kind of a tropical bird? Well, we don't know because nobody really followed. Well, maybe someone does know. I don't know. So that's a marvelous story.
Yeah.
And it's just, I'm wondering, I know, but some of these stories get lost if someone doesn't tell them. Because this was only, John Enders talked about, I don't think he ever wrote it up, to a dinner club called the Cambridge Scientific Club that meets the first Thursday during term at Eliot House at Harvard. But we have some people from MIT. I think we do. Anyway, so you better ask me a question or I'll wander out to infinity.
So these zoonotic diseases that you've been talking about, the zoonotic diseases that you've been talking about between the plague at Athens and then later on COVID is explained largely to be a zoonotic disease. Do you think that zoonotic diseases are kind of the major problem with a lot of these pandemics?
Where else would they come from?
Well, there have been theories, of course, that COVID came from a lab, that it was engineered. What do you think about ideas like this?
Well, certainly the plagues before labs existed did not come from labs.
Of course, right.
So that proves that they can happen without labs. As to where this particular one came from, that's a question for science, not for politics. Unfortunately, some of the people who were interested in it are interested in it for political reasons, and that will only cloud their judgment, and that's too bad. But there are, I hope, some people who are interested in it for scientific reasons, because it's an important scientific question. I myself do not know enough to have an opinion. I don't know.
Okay.
So
I feel like you're in this very interesting position during this pandemic in that you had worked a lot with policy in the past. You worked with biological weapons, and then of course you discovered mRNA, which is now the source of many of the.
Well, there was also somebody named Sidney Brenner, someone named Francois Jacob. I was very much the junior partner in that.
Okay. But so how does it feel to be in this unique position during this period?
Like, I better find something else nice to do.
I guess that's fair.
And I have, with a study to find out if we can conquer and avoid all future pandemics driven by aerosols.
Yeah. So I guess we can kind of...
And I'm interested in some other things too. I'm interested in the possibility of curing solid tumors, cancers. Not the blood cancers, but the solid ones. In bacteria, we have something called the SOS effect. It means if you irradiate bacteria with a very low dose of ionizing radiation or of ultraviolet light, they die. If you increase the dose, they don't die. The reason is that the enzymes that are needed to repair DNA breaks, which if unrepaired are lethal, the enzymes are not being made all the time. They're inducible. And these clever bacteria don't bother making those enzymes unless they get a little bit of radiation. But if it's too little, it's not even enough to induce those enzymes. And if it makes any double-strand breaks, even one, the cell dies. So the odd survival curve you get is that as you plot dose that way and viability this way, at very low dose, it's a very steep fall off. You're killing at high efficiency. As you increase the dose, they're not dying anymore until you get to very high dose. Yeah, that'll kill them. Now, do people have the same situation? Today, the radiation oncologist gives you a dose. They measure them in a thing named after a man named Gray, what's called 70, 80, 90 Gray. That causes hundreds of double-strand breaks and kills all the cells, almost. And so the oncologist comes back after this. He says, we did it. The tumor is gone. We killed it. Go home, have a good life. 2 years later, I'm sorry, it's come back. And it's a lot more aggressive. There's nothing we could do. You have 6 months left. And that's because at very high dose, yes, most of the cells die, the tumor shrinks. A few cells don't die, but they've got so many double-strand breaks that the pieces of DNA are reassembled at random. The cells are completely screwy. And out of these many really bizarre cells, there's some that have none of the mechanisms that respond to keeping a cell from reproducing endlessly, that tell a cell to stay in your place and don't become a cancer. They've lost that, and you have a cancer. So what if for the human you gave only 2/10 of a Gray, or 1/10? That would be enough to make 1 or 2 double-strand breaks. If we are like bacteria, those cells would die, because even 1 is lethal. But the enzymes that repair have not been induced, if we're like bacteria. And so the break persists and kills the cell. Either it or its offspring dies within a couple of generations. So that would be, it wouldn't work on blood tumors because you can't irradiate the whole body. Well, you could, but I don't know. That's another subject. There'd be problems. You hit the white cells and you get anemic and all that. But for solid tumors, even if there's more than one, course, if there are hundreds, then forget it. But maybe this would cure them miraculously. So I want to do that. I don't work with mice or rats, but I have a friend at Mass General. And the idea is we will transplant human tumors into mice or rats, and then with a control group and one that's irradiated, see if we can cure the solid tumor with a very, very low dose of radiation. Very low. So I want to do that. And now still in the area of experiments, although I wouldn't do that one. I've never done a thing with a mouse. I've never even killed a mouse. I've put cheese in a trap. That's as far as I've ever gone with a mouse.
Yeah, I'm curious. Have you looked at other forms of radiation? I work with alpha radiation a lot of the time.
Well, the things that are mainly used are gamma rays.
Right.
That's because that's easy. That's what X-ray tubes, which are called Coolidge tubes. They used to after a guy at Harvard named Coolidge. But now there are others. There's a proton beam, which is nicer because you can focus it and all that. But most hospitals don't have proton generators. Some do. It's a big deal. You have to have essentially like a cyclotron. Yeah. And I think that's all that's beamed these days. Well, there's sonic irradiation, but that's different.
There's carbon beams as well. This is actually my thesis.
There's what?
There's carbon beams as well.
Oh, yeah. Yes, big, heavy nuclei beams. Yeah. I don't know how those are used medically. Are they?
They are. Not in the United States, but in Japan they have, I think, 6 machines. I just had someone on who did this.
Why don't we use them?
They're still being tested. So it's a political thing.
I see.
Yeah.
And that's even better than protons, because they can be aimed very precisely.
They have a different Bragg peak is the other piece. So you have a different level of precision.
Very good.
Yeah.
Anyway, the other experiment, we don't understand aging. If you plot, there was a mathematician in England named Gompertz, Benjamin Gompertz, in the 1800s. He was a decent mathematician, not a great one, but he was a member of the Royal Society. And an insurance company, a life insurance company, wanted to have a better knowledge of the distribution of lifespan of its clients, because the premium that they charged in order to make money rather than lose money on life insurance policies, they needed to know the lifespan of their clients. So they hired Gompertz, and he got a whole bunch of data, because even then the census was done on males in England and Wales, not women, but men. I'm not sure of that. I'm pretty sure of that it was only men. And he observed that beyond a certain age, the death rate is a pure exponential. So that if you plot age this way and the death rate, which is the probability of dying, this way, you get a distribution that looks like this. At birth, the death rate is very high, but that's mostly malformations. And in those days, I suppose, also all kinds of infections. Was it before or after Joseph Lister? I don't remember. Anyway, and then it levels out and it's sort of flat, which means these are little babies who are dying randomly in time, falling out of the crib. But the death rate's very low at that young age. And then it goes up and levels off again. Teenagers are dying mainly not from any form of aging but mainly disease and accidents. And then finally, it takes off. And now the interesting thing is, he didn't know this, but we know this now. The slope is exactly a doubling time of 8 years. So if you do this with men who were born in Mexico in a certain year, you get this curve. And the slope, when it finally becomes a pure exponential, it's doubling every 8 years. If you do the same thing with Swedish women, doubles every 8 years once it gets into the exponential phase. Yet we all know that Swedish women live a lot longer than Mexican men. The only difference is when it starts.
Interesting.
Something triggers it at a much younger age in Mexican men than in Swedish women. And I just took those as an example, but there's nothing special about Sweden or Mexico.
Right.
It's true in all animals that I've looked at. The little things I work with, Deloid rotifers, it's true for them too. At room temperature, the strain I work with, once they start aging, it doubles every 3.2 days, or 2.3 days. So the question is, what is it that determines when this exponential process starts out? Because that's aging. Nothing else is aging. Because aging means an increase in the rate of death with time. It's like a trigger. Why? Nobody knows. There's a vast literature about this. No one's got an answer.
That's really fascinating because I've had several longevity people on and interviewed them, and
they may not even know about this.
Exactly. I've never heard about that study before.
I won't name names, but the longevity people you're probably talking about.
Okay.
So we need to understand the Gompertz phenomenon. There's no question of its existence. It's true. I've plotted this out for many different species of animal. Worms, mice, Deloid rotifers. Other people have done it for humans. So what's that all about? Now, there's an old hypothesis, the hypothesis of oxidative damage. And I don't see how it could give you this abruptness, but at least it's a hypothesis, and I have a way to test it. There are hundreds of papers about this. They neither prove it nor disprove it. I have an experiment which could disprove it. Unfortunately, it couldn't prove it. My little Deloid rotifers, which are little freshwater aquatic things that I began to study a long time ago because I was interested to know, why does sexual reproduction exist? Why doesn't everything reproduce asexually? It's a deep question. There are very marvelous answers to it. A lot of different answers. Good answers. So, they're incredibly resistant to ionizing radiation. You can put in dozens of double-strand breaks, and they fix them all up. There's no other species of animal, certainly not the tardigrades for whom this had been claimed wrongly, that are so resistant to radiation. And the hydroxyl radical is the most electron-hungry free radical that's ever exists in living material. Wherever a hydroxyl radical, which is oxygen with one electron missing, so it wants desperately to get that other electron so it can fill the outer shell. So it will steal an electron from anything that it comes in contact with. So if it sees an OH group in an aldehyde, in an alcohol or in a hydroxyl or in a COOH acid group, it takes an electron away from the oxygen, and now you have an aldehyde or a ketone, which means that that protein, which used to be soluble, is insoluble. And all kinds of bad stuff. It will aggregate, bad, not good. So maybe if the oxidative damage hypothesis is right, and to explain the immense radiation resistance and resistance against protein carbonylation as well, because we've studied both, we would find that when Deloids finally enter this aging stage of their life, they no longer have radiation resistance. They no longer have the ability somehow to prevent hydroxyl radicals, which are made from water and photons, no matter what kind of other chemistry there is. That's pure physics, Compton scattering. So if we should find, for example, that once the rotifers are in this aging phase, and I should have added that the rotifers we studied were young rotifers. They're the ones who are resistant to radiation. We never looked at aging rotifers. So if aging rotifers are aging because they've lost protection against free radicals, then when they're finally in the aging phase, they should also have lost their resistance to ionizing radiation, which we never measured on aging ones, only on young ones. So the experiment is to get a bunch of aging Deloid rotifers and measure their resistance to ionizing radiation. Very simple experiment. But we haven't done it. And that's the other. Those are the two experiments: the low-dose radiation on solid tumors and the aging thing. And I'd be very pleased if I could get those done. But the other things that I want to get done, I want to communicate with Elon Musk, and I don't know how. I've just really recently decided I want to do that. He's very interested about existential threats. So are a lot of people. But he has a lot of money. And he wants to spend it to avoid any of the really bad outcomes that could happen here on planet Earth by having another human population on Mars, and maybe even additional ones somewhere else. I don't understand why he also mentions Jupiter. From what I know about Jupiter, it's really difficult.
He's probably talking about the moons. A lot of the moons,
You're right.
Around Jupiter have very
right.
exciting
He's talking
scientific
about
process.
the one that begins with E.
Europa.
Yes, there are threats. We could have a meteorite wipe us out like it did with the dinosaurs. We could just have a terrific nuclear war, reducing our civilization to one so primitive that we could overheat the planet. But there's another threat that nobody talks about. We act as though the human genome were fixed. Well, already from the fact that there's so many different genotypes, we know it's not fixed. Because we know if the people who have genotype X have a lot of children, and those who don't have X don't have any children, that on average the human genotype will have changed. It's changing all the time. Okay. Now, most of the things we do aren't going to change it very much, very fast. Though we need to think about that. It gets you into some difficult political areas. And I don't have an answer to that. But not now, not soon, not even fairly soon, but eventually we're going to be guiding our own heredity, or at least we may be, because of CRISPR and things beyond CRISPR that have yet, though CRISPR itself is pretty easy now. And there are all kinds of refinements on the original CRISPR. So when we start replacing bad genes with good genes, that's probably safe. If I had a gene for hemoglobin S, sickle cell, and you could replace it with a gene for hemoglobin A, I see no danger in that if there are no off-target effects, and we think we've got those conquered.
There have been concerns about having certain groups of populations that have access to preventing all of these inheritable
kind
diseases.
Of political, social links.
Exactly, yeah.
But I'm talking about something different. When you want to, however, bring about some kind of enhancement, which doesn't mean replacing a known bad gene with a known good gene, because the known good gene is in nearly everybody anyway. So it can't be doing anything worse than what it's already doing, because it's already there. But if you put in a novel gene, and you know for sure the thing that it's going to do that is what you want, maybe it'll make my ears longer. You know, I want children with long ears. The question arises about what else might it do? Right. That brings us to something called the interactome.
Right. Transcriptomics can get very complicated.
Most genes that code for something code for proteins. But most of those proteins, whatever is their job, don't do it as monomers. They do it as dimers or trimers, and not always with homodimers or homotrimers. but with heterodimers and heterotrimers. That is, a given protein from a gene, depending on what cell we're talking about and what's the stage of that cell and what's the environment, what's happening. A given protein may be binding to, protein A may be binding to protein X to do whatever evolution and selection has taught that cell to do to have a maximum fitness. So that means if I put in a novel gene, maybe I know enough to say it'll do the thing I want it to do. But how many other configurations are there of that protein that we don't even know what they are or what they do? So you might lose things. Okay. What's the most precious thing we have? You really don't want to lose it. It's our humanity. What's that? Oh, well, kindness? Curiosity? Sense of awe when you see the stars in a town where there's no smog? Teamwork? We're not sure. The ability to be thrilled when you hear a Mozart Requiem played? We don't even have agreement. But in some sense, we know that we value our humanity. The most scary science fiction movies and science fiction books bring into your imagination things that are almost human. That's really scary. Some great lizard thing that's not at all human and just likes to eat people, okay, that's scary. He might eat you. But somehow, something that's evil and almost human, it's really scary. And I think that's linked to the value we place on humanity. And there's deep evolutionary reasons for that. There's a great book called The Price of Altruism. It's a very interesting book. I could describe it, but so this is a long research agenda, but the first thing is we need to understand what do we mean by our humanity. Professors of physics and chemistry and math, they don't know. Who knows? Well, nobody. Maybe poets? I don't know. Composers? Philosophers? Okay, so let's say they come along and tell us kindness is one. Maybe even physicists know that. I don't know. Although Pauli, if he liked to chew on ladies' necks from, didn't know about that. So, and then where's it determined? Well, not in the elbow, up here. Well, we don't understand the first thing about that. Could you even begin to study that now? Yes. In a very crude way that I hesitate to say because it will mean they'll get a grade of F from people who are watching this. But let's say that we get 10,000 people and we've got the full genome, and that's no problem because there's already millions of people who have the whole genome sequenced. So we have the full genome of all these 10,000 people, and we play the Mozart Requiem. And we ask, if you found this stirring, please raise your hand. And then we have this terrific computer, maybe it's even a quantum computer. And we say, computer, what is it in the genotype? Because although we don't know where kindness is in the brain, we know that the brain, except for things that are determined after birth, is determined by the genome. So computer, find out those polymorphisms, those genetic characteristics, which are in common amongst those people who were stirred by the Mozart Requiem. Well, maybe there are people who don't like the Mozart Requiem, but they like jazz. This gets you into a big cultural argument. My daughter thinks there's a nicer word than egotistical, but it means that my culture is better than your culture. So I don't know, but you could at least begin to find out. What are the genes that have anything at all to do with human emotions by doing this, which is called whole genome association analysis? And it's already being done for diseases.
Right.
Because it has a way, there are many diseases which are called by single mutations in so-called Mendelian genes. I don't know of any non-Mendelian genes, but all right, they like to call it. It's, anytime they want to mention Mendel, that's good, because he was very important. This idea of Mendelian genes, that really rubs me the wrong way. Could you please show me a gene that ain't
Mendelian?
What do you mean by it?
Yeah. So I'm kind of curious about this because recently I've learned how much information is not just in the genome, but also in the epigenome and in the transcriptome. Maybe. And so how much of this do you think would have an impact on a lot of
But that's all right, because we got whole human genomes where we know all the methyl groups are. Because you can do that nowadays.
Yeah, but a lot of the methyl groups will change over time. Or like my taste in music will change over time.
You're right.
My genome won't change over time.
I'm very skeptical of all this epigenetics. There's some. And to the extent that it exists, it's programmed by the genes themselves, by the sequence by the nucleotide sequence. It doesn't come from nowhere. And although Lamarck was actually a very great biologist, they don't come from reaching for the high bananas on the tall banana trees. That's not what determines DNA methylation, I do not believe. By the way, Darwin was even worse. Darwin hid his own defects very skillfully. He was a great, great guy. So was Wallace. But Darwin believed in the inheritance of acquired characteristics. But he not only believed in it, but he argued for it on the basis of things that he must have known were nonsense. I could give you some examples. But anyway, so even if it's right that there's a lot of inheritable, see, the stuff that's not inheritable we don't care about. If there's a lot of inheritable chromatin change, DNA methylation, any other secondary modifications, is very much open to question. But even if there is, I doubt that any of it, or at least much of it, is determined environmentally, but rather is determined by a program that itself is contained in the DNA sequence.
But do you think a lot of this humanity is heritable? Do you think a lot of these ideas behind humanity are environmental, or do you think that they're mostly from the genome?
I think that there must be. Well, here's why I think that must be heritable. Here's this building. I may know enough to, I don't know enough to do anything in this building. But there's some guy who knows how to make tables, and some guy who knows how to make bottles, and some guy who knows how to make doors and lights, a different guy, and carpets. You couldn't have this building without cooperation of the most intimate kind between different people. A long time ago, you probably couldn't catch a really large animal to eat it unless you had hunting groups, not just individuals. You couldn't begin to do all kinds of things that increased fitness, that is, reproductive success, without people cooperating with each other. So in some sense, part of what it is that makes our humanity is kindness. It's very hard to see how you could have had this degree of cooperation between people without the catalyst of kindness. If I knew that after we got the boar trapped and all sliced up, that you were going to take all the meat for yourself and not share any with me, I'm very much less likely to go on the next hunting trip with you, because I know you're going to try and get all the meat, not just... So this all works to increase fitness by expressing kindness. Cooperation in taking care of children. within small human groups, ensuring food supplies. But now there's something else that goes on. Because men can have children from many different women, and women cannot have children from nearly as many different men. It means that somehow, linked to being masculine, there must be behavioral programs that result from the greater competition that must exist, or at least a different kind of competition between men, a more violent kind of competition, the competition that wants to possess, just as you might want to possess physical objects, possess women and not have other men possess those women. That means, I think, a path to all kinds of hostility directed at other men. But there are programs to keep it from going completely bizarre, because you've got to cooperate with these guys too to build this lousy building that MIT owns and should sell. If the MIT Corporation is listening, you're losing money on this place. You should tear it down and put in very expensive, with concierge and everything, apartment house. Anyway, if those programs in the human brain of men go a little bit haywire with age, because lots of things go haywire with age, you might explain why it is that some men, not the majority, but develop very weird ideas when they get old. That other man is making time with my wife when he's actually not. This kind of paranoia that affects men more than women, usually taking the form of, my wife is making out with someone else, or it's not hers, it's this guy. We all have friends who, when they get old enough, have some crazy ideas. So there are these opposite acting forces on us because of the need on one hand to cooperate and because of the asymmetry between eggs and sperm. It's all very interesting. And we don't in any way at all fully understand it, but we can understand it in broad outline. So we don't want to start fooling around with the human genome, because both of these things, both of these programs, are very important, and if modified in some uncontrolled way, would have consequences we can't predict yet. So we want to be really careful of not losing our humanity, and especially because our humanity is delicately balanced between opposing things. So that if you tilt a little bit that way, you might end up with something you'll be very unhappy to have. So that's why I want to reach Elon Musk, because he's very much thinking about the future of the human species, way beyond his lifetime or any even much more distant time. He's thinking about that.
I know Max Tegmark, who I know you're familiar with in his Future of Life Institute.
Does Max know?
Yeah, Max has a connection with Elon.
Oh, well, I know Max real well.
Yeah, so I mean, if there's any way I can be involved in that, I would be thrilled.
Well, I got this prize from Max.
Right, the Future of Life Institute prize.
Yeah, and every time I have a birthday, he sends me a birthday card.
Right, yeah.
Thank you, I'd forgot about that. That's what I'm going to do.
Yeah.
So anyway, I want to get thinking started about this, and how do you do it? Well, getting in touch with Elon Musk is one thing. What he could do about it? Well, he could begin to at least encourage some other people to think about it and fund them so they could eat and have a roof while they do it. But there's also something else that can be done. There's always amongst the students at a good place like MIT or Harvard who are motivated, you could say, by philosophical questions rather than wanting to start a startup or because, gee whiz, it's really neat to work with electricity. But I want to know why things are the way they are. So up until about 1960, we had no idea how life works. How do you take the atoms of the periodic chart and put them together so that you got something that's alive? Until we got the DNA structure. And from that structure alone, there was at least a hypothesis how it could duplicate, how it could carry information, how that information could change mutation and crossing over. So for the first time, we could have an answer from science to the really much more philosophical question of what makes something alive. So the students who we used to get in the 50s and even the 60s, not all of them, but many of them, I would say were motivated by the philosophical content of the subject of biology. Now, it's like a big river which is divided into many, many little teeny rivers. So there's nice work being done in my department, and leaving out the brain science people, none of it is that fundamental. Interesting and good and high quality. Maybe there are a couple of people who are doing things with deeper meaning in the area of evolution. But there's something else. It's not a department yet at Harvard called the Origin of Life Initiative. Do you know about it?
Yeah, I've heard of that before.
And that attracts students from all different disciplines who have this philosophical. Because once we understand how life originated, all the philosophy about it will be gone. All the exciting thing about it will be gone. And the people who study it will just be good engineers. But the people who are interested in it now, they're this special group of people who are motivated by the fundamental question of, why am I here? Stop the train. I want to get off until I figure out what this is all about.
So I also want to kind of go back to when you were a graduate student and worked on the Meselson-Stahl Experiment, which has been described by some people as the most beautiful experiment in biology and describes the structure of how DNA replicates. Would you be able to tell me some of that story? Because it's a really cool story.
Well, like nearly everything I've ever done, it was an accident. In this case, the accident starts
a lot of happy accidents
in a swimming pool.
Okay.
Because sometime near the end of high school, I decided that I wanted to figure out how life works. So I went to my high school registrar, since I'd taken all the courses required to get a high school diploma, and I asked for my diploma. And she said, the law in California requires you have 3 full years of physical education, and you have had only 2. You have to spend 1 more year in high school. And I said, well, you mean just taking gym? She said, no, you could take the courses you liked over again. Well, so suddenly this was coming from a world in which everything was done for me. And now I have this crazy situation, which made adults who seemed to me before that very wise and everything was planned rationally, completely mindless. They're going to make me go to school and just run around the track for a year. So I began to ask, what can I do? And someone told me about the University of Chicago. It would take you without a high school diploma. So I went there, thinking I'm going to study physics, chemistry, and math. And it turns out you can't. Robert Maynard Hutchins, the chancellor, had abolished those courses for undergraduates. They still had them for graduate students. Instead, we're going to study the classics. We're going to read Plato. We're going to read Huygens. We're going to read Aristotle. We're going to read Harvey on the circulation of the blood. We're going to read Mendel. But we're not going to study things just for facts.
Okay.
So I was very glad we did that, and I read stuff I never would have read, especially in the field of world literature. But now, after 3 years, I'm done with that and wondering, what am I going to do with myself? So I took a year out to try to figure that out in Europe. And I was also sort of chasing a young lady who I knew was going to go to Paris. So when I got back, I started at Caltech. I grew up in Los Angeles, and if you grew up in Southern California, Caltech seems very, very much present, because there it is in Pasadena, and seemed to be kind of the best place in the world. So I went to Caltech as a freshman, and I didn't like it because everything was taught by rote. For example, the formula for the conservation of angular momentum. I was willing to accept the conservation of momentum on faith, because that's a physical principle. But I wanted to see how the conservation of angular momentum could be derived from the conservation of linear momentum. And that's not the way it was taught. Instead, we were taught as a separate equation. And there were many examples of that, except in one course, which was introductory chemistry, where Linus Pauling, using a very kindergarten variety of quantum mechanics, tried to teach us what is a chemical bond, sharing electrons in the outer shell. And that course I loved. So I left Caltech, though, because everything else seemed by rote. And besides, I'd lived a year in Europe, and these boys were none of them had been in Europe. They're not aware of what's happening in the world, somewhat younger than I am. So I went back to the University of Chicago to take those courses that undergraduates were not allowed to take. I took physics, chemistry, and math. And Fermi was teaching physics, and Urey was teaching physical chemistry. And they were a very good time for 1 year. And then I heard that there was, at Berkeley, a new program called biophysics. And to me, that was beginning. Bio and physics. So I enrolled in the physics department at Berkeley. And then I realized that I wasn't going to be able to get a PhD in physics, because being a state university, they had to admit nearly everybody who had a certain academic level of achievement, which was huge physics department. And they knew that they could only have a certain number of PhDs a year, dependent on the number of professors. And so they had to ruthlessly weed them out. And I knew I wasn't that good. Besides, there was no bio in it. But I drifted into a part of the physics department that had some bio. But to my disappointment, all that it was using ultracentrifuges to detect cholesterol in human blood. It was the beginning of this whole business. But the guy who ran that little department of I forget what it's called, biophysics, I guess, said, you should go back to Caltech. Well, I didn't want to go back to Caltech. I wanted to go back to the University of Chicago, because there was a program starting up called Mathematical Biophysics. So those are all the right words. Not only that, but it was headed up by a man with a big black beard and another man with big, thick eyeglasses. All that seemed very intellectually attractive. But my family is still living in Hollywood. And the year that I was at Caltech, I got to know Linus Pauling's youngest children, Peter Pauling and Linda Pauling. And Linda, I think it was Linda, not Peter, invited me to a party at her daddy's house, which was up in Sierra Madre, with a gorgeous swimming pool. It was a daytime swimming pool party, and I'm in the swimming pool. And out comes Professor Pauling. He knows me because when I was a freshman that year, I'd done a little research program on hemoglobin for him. So I'm in the water, and the world's greatest chemist is standing there wearing I think he was wearing a vest and a necktie and a jacket. And he looks down at me, and I feel like some kind of aquatic insect in the water. And he says, well, Matt, I have to paraphrase this, nice to see you. What are you going to do this coming year? I'm looking up at him like a god. I'm going to the University of Chicago to Professor what did I say his name was? Not Warshawski. I already said it once, didn't I? It's program in mathematical biophysics. Reshevsky. Pauling looks astounded. He says, and these were his words, "But Matt, that's a lot of baloney. Come and work with me, my graduate student." So I did. There was no paperwork, nothing. Okay, so now I'm a graduate student, and I'm taking Linus's course, The Nature of the Chemical Bond. And that week, the subject was the hydrogen bond. And the take-home problem was, what is the ratio of the strength of the hydrogen bond to that of the deuterium bond? In other words, the amount of work you have to do to pull it apart. And it turns out that you can get the answer of this ratio of work from the quantum mechanical zero-point energy. It's very beautiful, very almost a one-line proof, which to me was just astonishing that you could calculate something about chemistry and something so simple as the strength of how hot would you have to heat water before the hydrogen bonds go, if it was D2O or if it was H2O. Zero-point energy will tell you. Whoa! So I have this in my mind all about deuterium, and since I want to get into biology, like many chemists and physicists, their first ideas are completely ridiculous. They're applying some part of physics or chemistry to biology in a way that will tell you nothing. So my idea was to ask, what if you grew up a mouse only on deuterium compounds? Heavy water, heavy wheat, heavy carrots, heavy everything. Because you can do that. You can grow plants on D2O, and you can buy D2O to drinking water. So you get a heavy mouse. And I found in the literature there was a guy who tried to do that at Columbia. Mouse died. But it wasn't clear whether it was because you can't make a heavy mouse or because there was heavy metals in the D2O that he bought. He was killing the mouse with arsenic or something. Oh, but there, for sure you could make heavy bacteria. That was known at that time. So I'm thinking about all that, namely about D2O. And I went to a lecture by Jacques Monod. Do you know who he was?
No.
Monod and Jacob and the pajama paper and the whole idea of how genes are controlled by repressor.
No.
They proposed to, well, the very important French biologists did a lot, got Nobel Prize for how genes are controlled, what turns them on and off. In this case, a repressor, which is a protein molecule, binds to the site of an enzyme, beta-galactosidase, and if it's bound there, you can't make the enzyme, and if you can get it off, the enzyme gets made, and a small molecule related to lactose pries the repressor off, allows the synthesis, and lactose is exactly what that enzyme is there to digest as food so the bacteria can live. So it's just little wonderful. And they figured all that out. And Wally Gilbert got the Nobel Prize partly because he isolates the repressor. Anyway, I'm watching this lecture by Jacques Monod from the Institut Pasteur in Paris. And at that time, the question was, when you add lactose or any other certain chemicals called inducers to a culture of growing bacteria, they make the enzyme to digest the lactose. That's very sensible, very wise of the bacterium. Don't make it all the time. It's a waste of your time. Only make it if there's something to eat that you need that enzyme in order to eat it. Very good. But is that enzyme always present in an inactive state? And all that the inducer does is to somehow tweak it, maybe add a methyl group or change its shape, or is it made de novo? And Monod suggested that the way to find out was to measure the osmotic pressure across the bacterial wall as when it's presented with lactose and when it's not. Well, that's based on what's called the Donnan equilibrium. If you have a lot of things that can bind water inside of a bag, which is semi-permeable, the thing that can bind water can't go through the wall, but water can. and you increase the number of things that can bind water, more water will come in. And if the bag doesn't stretch, it'll break. But if it can stretch, it will swell. So Monod's experiment was, you add the inducer and see if the bacteria swell. Or in some other way, measure the osmotic strength across the bacterial membrane. I'm sitting there in the lecture room thinking, what a stupid experiment. How do you know there aren't holes in the wall? Aren't there thousands of other... But now I'm thinking about heavy water. That was all week long I'm thinking about heavy water. And it occurred to me that the right way to do this experiment is to grow the bacteria in heavy water. There was a German paper already that showed that could be done. And then add the lactose and quick centrifuge the bacteria down and resuspend them in light water. So anything that's made de novo, synthesized, before you added the lactose or the inducer, will be made with heavy hydrogen, deuterium. But if it's made afterwards, you've spun the bacteria down and washed them, and now they're in ordinary water, would have ordinary hydrogens. So that means you need to find out whether the enzyme activity has a high density or a low density and some kind of a density gradient. And in those days, we were using sucrose gradients for various purposes in centrifuges. And so the idea was, in fact, I don't even think I was thinking about a gradient then. Probably I was thinking about a tube with a sucrose solution of a known density, and you put this juice, ground up bacteria on the top, and now if the enzyme goes to the bottom because it's made out of heavy hydrogen, or stays up at the top because it's made of light hydrogen, that's what you measure. Where is the enzyme activity after you centrifuge it in this sucrose solution whose density is adjusted to be just halfway between the expected heavy and the expected light. And so I went to Linus, because I was doing X-ray crystallography, and told him about this idea, and he said, good idea, finish your X-ray crystallography. So I never did that experiment, of course. But then, a year later, I wanted to get more idea of what I could do when I got my X-ray crystallography done in real biology. And I went to see Max Delbruck, who a very famous man. He had been a student of Niels Bohr in Copenhagen, who got into biology as a young man. And he was in the biology department, I'm in the chemistry department. In those days, the mathematicians looked down on the physicists looked down on the chemists, and the chemists looked down on the biologists. Not probably the professors, but the students.
I think that still happens a bit today.
So there was very little communication between us chemistry graduate students and those biology graduate students. Dealt with slimy things. But I thought, since Max Delbruck is in biology and he's a physicist, he'll tell me what I should do with my life. So I go see him. First question, he says, what do you think of these new papers by Watson and Crick? And I said, I had not heard about them. He got up out of his chair and he had a whole stack of reprints of both of their articles, the structure in April 1953 and the biological implications in May. And he threw them at me. And they sort of fluttered to the ground in his little office. And he shouted at me. He said, get out! And don't come back until you've read them. Well, I heard come back. So I read these papers, tried to understand them. And then he told me that this mechanism of separating the chains, each one makes a new one, can't be right, because these DNA molecules are very long. And to separate the chains, you have to rotate either the mother helix, or even worse, swing those arms around for as many 360-degree turns as there are turns in the whole DNA molecule, which is very long. And hydrodynamically, you cannot do it. Therefore, there have to be breaks. And the Watson-Crick scheme for replication has got to be wrong. You have to break it and then reseal them, because then the rotation is only around the single bond of the chain you didn't break, and then you reseal the break. And the particular model he had for this exchanged pieces between, you start with one molecule, end up with two. The matter of which the original mother molecule is made is distributed half in this daughter and the other half in this daughter. In other words, you get 2 half-heavy ones, but then later you get 4 quarter-heavy ones, and then later you get 8 eighth-heavy ones, and so on. In other words, the mass of the parent is being diluted out indefinitely as replication proceeds generation after generation. And there was another hypothesis that one double helix does some kind of a magic without separating the strands at all, and by some weirdo non-existent quantum mechanical effects causes the organization of an identical double helix nearby. And there was actually a guy named Herbert Yale, was a kind of a physicist, who believed in that. And in those days, there were some arguments you could make for it. It all depended, well, I won't go into that. So sitting there, the second time I went to see Delbert, it dawned on me that the right way to find out about this is not Monod's osmotic strength experiment, but it's the experiment I thought of for telling where the beta-galactosidase, when the beta-galactosidase is made, applied to DNA, however. So I had this idea. Again, I go to Pauling. Good idea, he says, finish your X-ray crystallography. At that time, I'm living in the Athenaeum, the Caltech faculty club, but graduate students could sleep in the upper rooms. And Jim Watson is there for that year. It's the year 1954. And we would go there. Dinner is at 6, but we'd get there early because we're hungry. And we'd sit in these big soft leather chairs and talk, usually about girls because there weren't any. And there was only one, in fact. Her name was Rachel Morgan. And Jim and I both dated Rachel, so we had lots of things to talk about. And I told him about this idea, and he said, no, it's a good idea, do it in Sweden. And I thought, and I'm still not sure, that what he meant was do it in Sweden because there are girls in Sweden, or maybe it was because that's where the ultracentrifuge was created by a man named, his first name was T-H-E, Svedberg. And that's the unit of sedimentation in a centrifuge, the Svedberg. Anyway, Jim thought it was a good idea too. But the next summer, the summer of that year, 54, Jim wanted me to come to Woods Hole with him to do a certain experiment with RNA, because Jim believed that maybe there could be a double-stranded RNA molecule just like DNA. He's always deeply interested in RNA. And there was an experiment done by a great British physical chemist who died much too young in a train wreck, named John Gulland. A very simple experiment. You take DNA and you titrate it with acid till the solution is very acid, like pH 1. And then you back titrate it by adding alkali till it's back to 7, which is neutrality. And he discovered that the amount of acid that you need to add to get back to pH 7 is much more than the amount of base it took to get up to pH 14. And there's only one explanation. There had to be a structural change involving the production of titratable groups caused by exposure to base. And that could only mean the breakage of a lot of hydrogen bonds so they could now be titrated. Things that were COOH, but where the hydrogen bonds between those groups kept them from being titratable by keeping them bound to each other. So he concluded from this that the DNA structure was a system, long molecules, because we knew that from the birefringence, somehow with a highly structured system of hydrogen bonds. Well, if RNA was like DNA, it should titrate in the same way. You add acid to get up to pH 1, and add base coming down. You have a pH meter. You measure the pH. You measure how much acid, how much base, and you find that you need, let's say, 2 moles or I wasn't working with huge amounts, 2 millimoles, maybe 2 micromoles of base to get down where only 1 mole. So I did the experiment, and there was no evidence whatsoever for hydrogen bonds. Jim was disappointed, but I met a guy named Franklin Stahl. And that was because although I was hired there as a teaching assistant so they could pay me, but it was a fraud. I was a laboratory assistant to Jim, but there was no budget for that. So he snuck me in under the door as a teaching assistant. I did no teaching assistants, but I met with Jim, and Sidney Brenner was there that summer, and they would get together and talk about how to run the class in the coming week. I used to meet with them in an upstairs room in a building. Have you been to Woods Hole?
I've not.
Oh, you should. Just get on the bus. It's 90 minutes. Anyway, Jim goes to the window and he points down across the street, sitting under a tree, sitting in the grass. He says, "See that guy? He thinks he's pretty hot stuff." I don't remember Jim's exact words, but let's see if we could give him the Hershey Chase experiment to do all by himself in one afternoon. See how good he really is. That's a hard experiment even for 2 people as skillful as Hershey and Chase, but to do it in one day and for one guy. So I felt sorry for this guy, and I went downstairs and I told him, they're going to gang up on you and they're going to ask you to do this experiment. And he said, he was a very good experimentalist. I'm not sure what he said, but I think he felt this would be no problem for him. And he knew what the Hershey Chase experiment was. But he told me what he was doing. I was interested in what he was doing. It was a certain experiment in radiation biology of bacteriophages. And that was the first time I really learned something about phages, because he's a wonderful explainer. And then he asked me what I was thinking about, and I told him about this experiment. So we agreed we'd do it together. And it wasn't until 1 year later he came to Caltech as a postdoc. And I'm still doing my X-ray crystallography, but since Frank had just arrived, I said, "Okay, now we're going to start the DNA experiment, right?" Frank says, "No. You have to finish your X-ray crystallography." And that tells you a lot about Frank's character. He's probably the most moral guy you ever met. And in his mind, and in fact he said it would be bad for your character if you didn't finish your X-ray crystallography. So I had to finish it, which I did. I never published it because I was eager to get out of that. And we did this experiment, and we did all kinds of things on the way. It's a long story, but there's a whole book about it by Frederick Holmes, who was a historian of science at Yale. He died much too young. But it's called Meselson-Stahl and the Most Beautiful Experiment in Biology. There's a videotape. If you put my name into YouTube, you'll find a videotape of Frank and me, both now really old guys. I think I'm already maybe 90. I think 90. And Frank would have been 91, sitting on a sofa, talking about all this.
I watched that to prepare for this.
Did you see it?
Yeah, it was a great video.
He had thought that this was the most beautiful experiment in biology, but then John Cairns had said that Mendel's work was the most beautiful, which it was. Mendel is, generations of students are dragged through reading Mendel or about Mendel and reading all the wrong thing. All they learn about is these meaningless ratios. 2-to-1 segregation. That's not the point. All that's because P's happen to be diploids.
Yeah, that's what I was taught.
Working with haploids, because there you don't have to worry about any of these ratios. And dominance and recessiveness have no meaning. The thing about Mendel is that he rescued Darwin. Darwin was in the dumps before Mendel was rediscovered in 1900 because everyone, including Darwin, believed in blending inheritance. That is, if a variation should occur in an individual, in his offspring, or her offspring, it would be diluted by half. And in the next generation, diluted by another half, and by another half. So that after a few generations, this characteristic, like, say, increased strength or something like that, would be diluted to such a small thing that it could not be recognized by natural selection above the noise level. So evolution would be impossible. Whoa. Unless, by some magic, the same variation occurred in everybody at the same time. And then you can intermarry all you want. There's no dilutant present. It won't get diluted because everybody's got it. Well, how can that be? Well, that could be if variation is caused by changes in the environment. So if there are enough people who are doing something with a hammer, their children will all have genetic changes that make them better at using hammers. And since there are a lot of those people, we don't have such bad dilution. There's still a problem, obviously, because not everybody is hammering. And certainly not everyone is playing the violin. But Darwin wrote a whole book about it, the variation of plants and animals under domestication, or at least the last chapters of that book. It's a crazy theory. If you read it, I won't bother you with it. The most insane speculations on the basis of the weirdest, shakiest, bad evidence and made-up stories. Right. But as long as you believe that, the only way that evolution could work would be if Lamarck was right, that all variation was caused by environmental change. So the important thing of Mendel is not those crazy ratios. It's not dominance and recessiveness. It's the fact that if you cross two purebred lines that are homozygous because you've bred these peas for generations and generations by selfing, let's say a white flower with a red flower, and you get a pink flower in the next generation. If you cross those with themselves, you get the nicest white and the nicest red, just like the distant parents. There's no blending. Right. There's still pure red and pure white. They emerged from this genetic mishmash, undiluted, unblended. It took a while for biologists to accept this idea that there's no blending. But that rescued Darwin, because without it, Darwin's whole edifice, if you believed in blending, Darwin has to be wrong. Lamarck has to be right. So it was really a big deal, and that's the way it should be taught. Well, we've talked a lot.
Yeah.
But there was, as I said, things can happen by accident. So the accident that I went into the swimming pool to Linda's party, which made me a graduate student of Linus, that was the accident. I would never have done that experiment with Frank Stahl if I hadn't gone to the swimming pool, if Linus hadn't walked out at that moment and said, "What are you going to do next summer?"
Yeah. So how did that end up going all the way to you getting involved in policy?
Well, I wouldn't have become Linus's graduate student. Yeah. I wouldn't have taken the course in the nature of the chemical bond. I wouldn't have gone to hear the lecture by Jacques Monod. I wouldn't have therefore thought of using density to label anything. I wouldn't have gone to see Max Delbruck. I wouldn't have known that there was a DNA problem. I certainly wouldn't have thought of density. Well, I might have, but that's not why I did. So you could all say it was the swimming pool. Yeah. Why not?
Let's make a good story.
Why not? The next accident. I'm between marriages. It's the summer of 1963. I have a friend, Alan Forbes, who owns the most beautiful little house in Georgetown. And nobody's going to be there in the summer of 1963. Also, Paul Doty, my friend, chemistry professor, no longer alive, at Harvard. He's on the General Advisory Board of the newly created Arms Control and Disarmament Agency in Washington, created by Jack Kennedy, partly in homage to Hubert Humphrey, who used to say that if he were elected president, which he wasn't, didn't get the nomination, but he would ask the government to study arms control and perish the thought, disarmament. So, for whatever motivations anyway, Kennedy created this thing inside the State Department. So here's this new agency wondering what to do with the money that the Bureau of the Budget had given it. And since it had just been created, it needed new ideas what to do with the money. Because if you give the money back, then in the following year, you don't get as much. So Doty told me that they were going to hire 6 people, academics, just to come in there and think up things for them to do. And would I like to be one of them? I said, sure. I had nothing else to do. Maybe I'd meet somebody who I'd marry. I had this wonderful house, guaranteed. I'd be the only one in it. So I went down there, and they said, you're going to think about European theater nuclear arms control. Well, you know, generally people who are scientists, especially if have even a slight little grain of physicist in them, have this arrogance to think that give me any problem, I'll be able to think of something. So I agreed to do that. But it gets to be very embarrassing because I know nothing about the subject. That's okay, but there are people who know a lot about it. Henry Kissenger had already written his book about it called The Necessity of Choice. And not only that, but very high-ranking people came to my desk to brief me. Llewellyn Thompson, who was our ambassador to Moscow, who was the number 2 at the Defense Department then. I'll think of his name.
But you and Henry Kissinger were quite close, weren't you?
Yes, but that's for another reason, and that predated this. But we weren't very close yet. We knew each other. I didn't really get to know Henry until around 1966, and this is 1963.
Okay.
So, I'm just very embarrassed. They expect me to write something at the end of the summer. They already knew more than I did by many orders of magnitude. But I'm a chemist and a biologist, so I went to my boss, who was a wonderful chemist from Montana named Franklin Ford. After he left the agency, he went to Cornell. I said, I'm a chemist and a biologist. Why don't you have me think about chemical and biological arms control? He said, sure, you can do whatever you want. We're all going to Moscow to negotiate the atmospheric test ban. And we had a guy who was thinking about that. His name was DeRuel. And just like you, he was a student at Caltech at one point. And he had a bunch of classified documents he'd collected. And they're in the safe, and you can have all those documents to get you started. Do whatever you want. So I said, okay, that's much more comfortable for 2 reasons. One, not many people had thought about that. And 2, I could understand it better. Or at least I had a credential that sounded more like. So I decided I'd go to the CIA and see what we thought other countries were doing. We all had very high clearance at the Arms Control Agency because the Congress was worried that people who were interested in disarmament must be a place where there would be communists. I guess that was their thinking. Because we all had to have the same very high, we all had Q clearance, which is for nuclear stuff, even though I wasn't doing anything nuclear.
Right.
But that was the idea, which meant that the place was more like a college than like a government agency. Because it meant that if you all had the same clearance, you could talk to the guy at the next desk or in the next room on the same subjects. The only other criterion was a need to know, which is a little hard to define anyway. I mean, I really had no need to know anything nuclear, but still, I could talk to my pals about nuclear weapons. Anyway, and then I thought I'd better know what we're doing. At the CIA, we didn't know for sure what anybody was doing. We had suspicions. We knew there was a lab of some biological kind in Sverdlovsk. We didn't know what they were doing inside. And in Fort Detrick, which was the main place where we did research to develop biological weapons, I was treated very well and given a long tour of the whole place by a very nice man who had been an assistant professor at the Harvard Medical School. He was a colonel. His name was Leroy Fothergill, a very nice man. And he took me all around. We came to this big building, and from a distance it looked like it had windows. But you get up close and you see there's only imitation windows. And I asked him, why is that? Well, we don't want people to know that we're... I thought that's about the most stupid thing you could do, because if you have a good telescope, you can see these aren't real windows, and therefore you'd say, my God, what are they doing in there? But that was it. So I said, what are we doing there? He said, well, we have a 10,000-gallon fermenter in there. We make anthrax spores. So I said, well, why do we want anthrax spores? He said, well, as a strategic weapon, it would be much cheaper than nuclear weapons, much cheaper than hydrogen bombs. It would save us a lot of money. I don't know if we had hydrogen bombs. Yeah, of course we did. They came in the 50s. So I'm thinking, and I must have had this idea for sure before I got back to the State Department, which is a long taxi cab drive from Maryland at Fort Detrick back to the State Department building. And it dawned on me, the United States is the richest country in the world, and we have nuclear weapons. Would we welcome the advent in the world of a 10-cent hydrogen bomb? Everyone could have one. Little children could have them. Everybody could have hydrogen bombs. Well, that's biological weapons. What Colonel Fothergill has just told me was not the reason why we want them. It was the reason why we should absolutely not want them. And not only that, it was the kind of reason that regardless of your ideology or anything, was inarguable. We do not want to stimulate interest in these things because all the countries in the world could have them. This is the kind of argument that if you could get it on the desk of the... There's only one person who could stop this program. It's the President of the United States. So if you could get this argument on his desk, that would it. They'd go away. He would surely stop the program. This isn't true in all the other things, like maybe you want to outlaw multi-cartridge magazines for attack rifles or something. Well, there are arguments on both sides. And not only arguments, but money, too. But this was an argument for which there was no comeback at all. You could have said, well, we'd like to study it so we might know about it in case someone else wants to do it against us. But that's not what we were doing. We were actually designing munitions. Bad. And doing open-air testing that other guys could know about. So how am I going to get something on the desk? Everybody in the world wants to get something on the desk of the President of the United States. How do you do it? I remembered, I don't know where I'd heard this, but an anecdote about President Roosevelt. That someone, I guess a banker, some banker friend of his, went to see him and convinced him of doing something. I don't remember what. But I do remember the anecdote that Roosevelt said to him, "You've convinced me. Now go out and put pressure on me." Meaning that it's not easy for a president to do just anything. There has to be a sentiment for doing it in the country. That means the media. In those days, that meant the newspapers. There was no TV. Or if there was TV, not many people had it. It meant that the Congress, if something was wildly unpopular in the Congress, no president can do it. It meant even his close advisors. Because if they're all saying, don't do it, or it's unimportant, don't bother, it's not going to get done. So in biology, when we do experiments, we prepare what's called a protocol. You have to list the reagents you're going to need, the equipment you're going to need. It's better to write this down before you do it, because otherwise, oh my God, I don't even have that reagent. You've got to have a protocol. So I wrote a protocol, because that's what I do in biology. So media, I have to get to the press. Congress, I have to get the House and Senate. Advisors, well, Jack Kennedy populated the government almost entirely with people from Harvard. I'm from Harvard. Just knock on any door. So I concocted this plan. How about the press? A friend up here knew a guy who was at that time managing editor at the Washington Post, Howard Simons. Howie had started out as a science reporter, had taken a course in physiology at Woods Hole, but he had become the managing editor of the Washington Post. So my friend up here, Howard Hyatt is his name, said, go see Howie Simons. He'll tell you how to get to the press. So I went to see him, and he took out another yellow pad. I still have it. And he said, I'm going to write down the name of all the science reporters of the major newspapers in the United States. In those days, most papers had science reporters. Nowadays, they don't have the money. But even now, of course, the Times does, the Post does, San Francisco Chronicle, a few papers. But in those days, there were about 30 names. He says, go see, or at least talk to all these guys, and you'll get a lot of press, because once a week, their editor comes to them and says, we got the science page in the weekend edition. We got to have something in it that will really get the readers excited. Biological weapons is pretty exciting. So I went to see many of these people. One of them just died, age 99 or something, or 100. His name is Perlman. He was at the San Francisco Chronicle. I went all the way out there to talk to him. I talked to the guy at the Baltimore Sun. Of course, I'd already talked to the guy at the Post. That was Howie. And I did this. And then in the Congress, I started out by, congressmen and senators have assistants. There's the administrative assistant, he runs the office. There's the legislative assistant. They worry about bills that are coming in and bills that their congressperson might want to write. And these are usually young people. So I could go down there and just introduce myself, because many of them are interested in finding good stuff to give their boss. That gives them, credit, makes their lives enjoyable. So I met a bunch of people. Some of them are still around. And then I began to meet some of the people they worked for, but especially two people. One was a congressman. His name was Richard D. McCarthy, but he was called Max. And he was from Buffalo. But he became very interested in chemical weapons. He wanted to find something that would make his name recognized. And at that time, there was a proposal to ship a bunch of obsolete nerve gas bombs across the United States from Colorado, where they were stockpiled, to New Jersey, where they'd be put on old Liberty boats, Liberty Hulks, sailed out to sea and scuttle them and dump them, get rid of them. Well, governors in between didn't want railway cars with these things going through their states. The newspapers got a hold of it. It became a great big deal. And so he thought that he'd become the leader in the Congress against this. And so I went and introduced myself to him, and we became great pals. He wrote a book, and so on. And through him, I got introduced to the chairman of the Foreign, in the Senate, it's called the Foreign Relations Committee. In the House, it's called something else, forget what. But anyway, I forget even who was, it was a man with a Polish name who was the chairman. I forget now who it was. But I got to know him. And over in the Senate, I got to know Fulbright. Have you ever heard of him? He was an historian, a very learned man from Arkansas. And he was interested in biological and chemical weapons for a reason it will take too long for me to explain, but he wasn't really interested in them for themselves. He was interested in them because he was against the idea of anti-ballistic missiles. He thought that the terror of nuclear weapons would maintain the peace. And if there was something that could prevent a nuclear-armed missile from reaching your country, therefore, these would not be peacemakers, they might be war makers. So he wondered whether biological weapons could somehow be used regardless of ABM. His ideas were not clear, but we became good friends, and it's a long story, but he was in a position as chairman to do all kinds of things. But then there's Henry Kissinger. So how do I know Henry? Well, first of all, my building at Harvard is called the Biological Labs, and next to it is a building that used to be called the Semitic Museum. It's now called the Mesopotamian Museum. And Henry Kissinger's office was on the ground floor there. During the war, that building was sort of taken over by government people. And on the third floor, there was a kitchen and two Hungarian ladies who made lunch every day. So you could buy your lunch there. And it was, like 5 orders of magnitude better than any of the food that you could get at the other Harvard canteens. And Henry would have lunch there, and I would have lunch. Occasionally I would talk to him. I think that's how I got to know him. But around 1965, I'm not sure what year, but he and I guess Carl Kazin over here at MIT created what was called the Harvard-MIT Arms Control Seminar. And it met, I think, every month at night. Though I think it was after dinner. I don't remember eating anything. Anyway, I have the minutes of the whole thing. And the first time I really interacted with Henry, he went to Vietnam in 1966. At that time, our ambassador to Vietnam was Cabot Lodge. And he had been wisely appointed to be ambassador by Jack Kennedy because Cabot Lodge is a Republican. And already there's a political hazard in appropriating money and sending soldiers, or in those days we used to call them advisors, to Vietnam. So it's good to have a Republican in on the game, so to speak. So Henry went out there at the request of Cabot Lodge to give him his opinion of what's happening. So Henry, being Henry, instead of just going to Saigon and talking to the higher-ups, went all over the place. There are 40 provinces. He goes to many of these separate provinces, spends 6 weeks doing it, gets everybody upset because he's asking all these penetrating questions. He comes back and gives us, at the arms control seminar that night, a talk. And he says that the authority of Saigon doesn't reach beyond Saigon. Each province, the most important man, is the one with the largest private army. The Saigon government is totally inefficient, corrupt, and the same is true in all the provinces. The enemy, the Viet Cong and the North Vietnamese, are much better organized, much more efficient. How naive I was. I have the transcript of the record. Little Mathew gets up and says, if they're very efficient and our side isn't, why don't we change sides? You might have thought Henry would say, you dirty, filthy little communist. That's not what he said. He said, we have nothing to offer them.
That's an interesting way to look at it.
Because they just wanted us out. That's what they wanted.
Right.
So that meant I could talk to Henry now. So we used to talk. And now we get flash forward some more, and Nixon is elected. And I'm going back and forth to Washington because of my campaign to get Congress, the press, government advisors interested in the issue of biological weapons.
How old were you at the time? How old were you at the time of this? I'm sure this is after graduate school.
In 66, I would have been 36, I guess.
Okay.
I was born in 1930, 36. So there used to be, I guess there still is, a ramp at American Airlines. I don't know if American is still right here, but it's a ramp that goes down to the gates. And I forget whether I was coming up or I was going down. I just remember it was a steep slope, and Henry's going the other way. And by now we know each other pretty well. I think we'd already traveled together in Europe because he went to Pugwash conferences. You ever heard of Pugwash?
No.
Oh, well, just before the death of Bertrand Russell, he and Einstein issued a manifesto which was calling upon scientists of the world to work for peace. And a very wealthy railway owner in Canada named Cyrus Eaton put up the money for annual meetings of very high-level Soviet scientists and American scientists and British scientists to meet. And he owned some kind of estate at a fishing village in Nova Scotia named Pugwash. And so for some reason, this movement was called Pugwash. And in those days, it still exists, but not in the same way at all. But in those days, it consisted of the most senior scientists from the Soviet Union and from the United States and Britain. The idea being that they could communicate directly with heads of state, or almost directly. So I didn't know why Henry was going to that. He's not a scientist. The reason, I didn't know it at the time at all. He had established a secret channel of communication with North Vietnam because of a bacteriologist named Herbert Markovitch, who was at the Institut Pasteur. Markovitch knew a mathematician named Raymond Aubrac. Raymond Aubrac, Ho Chi Minh, when he was a student in Paris, kind of down and out, lived with the Aubracs, and I think was the chef in some kind of restaurant and earned a little bit of money. But he lived with the Aubracs. So here's a Frenchman who knows Ho Chi Minh, and another Frenchman who knows Aubrac. I don't know how Markovitch initially contacted Henry. I better find that out. It's known. But anyway, Henry would come to the Pugwash conferences, I think, as a cover, because his meetings in Paris not with Ho Chi Minh. Ho Chi Minh sent a man named Le Duc Tho, who was, I guess, like foreign minister of North Vietnam. But they had meeting after meeting, and that's why Henry and Le Duc Tho both got the Nobel Peace Prize. Le Duc Tho refused it because the agreement, the ceasefire, didn't last. Anyway, while Henry was going to Pugwash meetings, and I'm going to Pugwash meetings, and I had a car and he didn't, we got to know each other then, too, even better. Mm-hmm. And so now on this ramp, anyway, we sort of bump into each other. Henry is sort of built like a fire plug. I mean, he's thick. I can remember this feeling of bodies smashing. And then he said, Matt, they call me Matt, what should we do about your thing? Because I had been through ACDA and all that stuff, and my thing was chemical. I said, I'll write you a paper. So that was the first paper I wrote for him. It was called The United States and the Geneva Protocol of 1925, which is an international treaty that we suggested and never ratified ourselves, that prohibits the first use of chemical or biological weapons. It allows reprisal. That's not retaliation. Reprisal means no larger an effort than what was done to you. You can't retaliate with a massive response. Reprisal is more limited than retaliation. So it allows reprisal in kind. But we had not ratified it. So I thought that was a reasonable thing to write a paper for the president, because I knew if I went for Henry, I'd go to the president, President Nixon. By the way, when this happened, Nixon hadn't yet taken office, but it was announced that Henry would be his national security advisor. So I began to write a series of papers for Henry and to go down there and talk to him. And I liked him, and I still like him very much. I just sent him a birthday card. His birthday was last Friday. He's 98, I think. I'm 92. And I had lunch with Henry last year. Or was it this year? I'm not sure. The weather was warm. It was spring. I guess it was spring of this year. I'm not sure what it was. Anyway. I used to see him pretty often. And I like him.
Mm-hmm.
I think he was always trying to contain the war. I think that Nixon was always wanting to expand it. And the people who Nixon was getting advice from was a very hawkish group. But I know Henry is, well, when he came back from that trip to Vietnam in 1966, he was very tired. He had just come in from the airport. And I don't know why it was, but we were having sherry in his office late in the afternoon. And I remember he said to me, he's just back from Vietnam, "Matt, now I know how the good Germans must have felt."
Wow.
Every once in a while I ask him what he meant by that. Twice I've asked him. And both times he just fell silent. And after a while it became embarrassing just to sit there in silence. So I resumed the conversation. If we meet again, maybe I'll have the courage to say, "Henry, why did you say that?" But you can imagine why that endeared him to me.
Right. That's a really powerful
and I
statement.
thought of Henry as the little Dutch boy. I don't know if you know this child's story.
I don't think so.
But there's a hole in the dike, and water is rushing in. It's going to drown everybody. The little Dutch boy puts his thumb in, because hydraulically it makes no sense. Mm-mm. You can't get that much water through a hole so small that you could put your thumb in it. Mm-mm. But it saves everybody. So what's he really doing, the little Dutch boy? He's stopping the forces of destruction by just being there and stopping. Is it possible that Henry saw himself? I mean, you got Curtis LeMay still in office, the guy who wants to bomb the hell out of Russia. You've got the most important senator, Richard Russell, chairman of Armed Services, who during the missile crisis with Jack Kennedy, it's all on tape. You can listen to it. It's on YouTube. "Jack, we might as well start it now because it's going to happen anyway," meaning the nuclear war. Does Henry see himself as a little Dutch boy? That to have any influence at all with these guys, he's got to be what's called a realist. I don't know. It's complicated. Yeah. It's complicated. I mean, the thing that really bothers you, of course, is Chile and Pinochet. And the fact that there's such things as this wonderful guitarist, Jara. Pinochet's police destroyed his hands before they shot him dead. Anyway. It was accident that I got involved in all this. But because I had in my lap an unbeatable argument, I had the responsibility. I had to. How could I throw this away? I had the time. I came from the right place. I had the prestige. I'm a famous scientist, even though I'm still pretty young. I have important friends, and I know how to do stuff, and I have good shoes. So that was an accident. Just like the same for the DNA.
So once it actually got onto the desk of the Nixon administration.
What?
So once it actually got onto the desk of the Nixon administration, what other hurdles had to be overcome?
Well, as I say, it's such an unbeatable argument that you have bureaucratic inertia. But for the United States to renounce something that it wished didn't exist, that doesn't. It's pure gold. It's wonderful. How could you argue against it? There were a few diehards who don't like treaties at all, but there was no real opposition at all. You had to convince the Russians. The Brits already had been thinking about it.
Were you involved in that process of the kind of international negotiation?
Yeah, very much. For a number of reasons. There's a man named Lord Solly Zuckerman is his name. And during the war, the Second World War, he's a zoologist, director of the London Zoo, on the board of directors of the London Zoo. Worked on primate fertility and stuff like that. But during World War II, the Germans are bombing London. And Quonset huts, things of that shape, corrugated metal, were built in London for civilians to duck into in case the sirens would ring. But their eardrums were being blown out. So because he worked with primates, he was asked, use primates to find out what's going on, because you can't do experiments on people. So you put a primate in one of these and you blow up something outside. Well, of course, it's a parabola.
Right. Yeah.
Almost.
So everything's getting
It's
focused.
focusing.
The noise.
It's okay if you're in the good part of but in certain parts, the sonic energy is enough to blow out your eardrum. And he figured all that out. So he then became science advisor to the Prime Minister through several British governments. And very high placed, mainly because he had a very close, in England, the private secretaries are very powerful. They're a civil service that does not change when the regimes change. It's unlike here. So the private secretary to the minister of this or the minister of that has almost as much power as the minister himself. So the private secretary to the prime minister and to the minister of defense, they were all his great pals. So I was able, through Solly, to be able to tell the British the state of play here. And when the British see that something is being taken seriously in the United States, they take it seriously because they're very dependent on us. They're dependent, and we are dependent on them too for intelligence, but they depend on us for intelligence too. And they very much value their alliance with us, of course. English speaking, similar values. They're exposed over there in Europe. We're this big arsenal. We saved their necks in World War I, even more so in World War II, all of that. So for that reason, I was able, I think, to play some role, but I'm sure things would not have been different. On the one hand, I was very active doing this, but I think if I wasn't doing this, things would have happened the same way anyway. Because the argument itself for banning biological weapons was so appealing to any big power, communist, capitalist, no matter what. It's just that big guys should oppose cheap weapons because they're big and rich. So there are two parts to this. Yes, I was very active through Solly and through my friend Julian Robinson, also an academic. We created something called the Harvard Sussex Program on biological and chemical armaments. But I think it didn't make any difference. So you can be very active on something and it doesn't make any difference. So I doubt that it made any real difference. Where it did make a difference is a problem in our government is there's so many problems, is getting your problem up at the attention level. And I think I was able to do that because I was able to go between agencies, whereas if you're in any one agency, unless you're very high up, you're sort of trapped there. You're not allowed to go around politicking in other parts of the government. I mean, you can see why that would be chaos. You might do it with your social friends, but not too conspicuously. But I was a free agent, so I could go see anybody. So I did. I saw Cy Vance, and everybody agreed with this argument. Everybody said, of course, it would be bad for the United States. So the only problem was to get it up high up on the agenda. And I think part of that was because of an accident. The Minister of Defense in Germany, we had stockpiled chemical weapons in France. And then de Gaulle wanted to get out of NATO. So we thought we better not stockpile our chemical weapons on French soil. And maybe de Gaulle told us to get them out. I just don't know. But anyway, we moved them into Germany. So that's where our whole stockpile of nerve agent chemical weapons for NATO is. And the German Minister of Defense's name might have begun with an H, I'm not sure, formally requests German authority to use those weapons. In other words, what's called release authority. Well, in the United States, release authority for nuclear weapons and chemical weapons was the same, and it was only the president. The only one who had the authority to order the use of nuclear weapons or the use of chemical weapons, President of the United States. So if we gave release authority to Germans, even though they're our allies, that sort of means, what about nuclear weapons? And nobody was in favor of letting anybody else initiate the use of our nuclear weapons, because that could mean everybody dies. But Germany is a good friend. How are we going to answer this guy, who it turns out was acting without the knowledge of the prime minister? That's a whole other story. Well, what do you do when someone asks you something and they're a good friend and you really want to say no? You say, oh, I'll think about it. We'll study it. Well, that meant that this little issue of biological weapons that only low down in the government was being thought about suddenly is a big deal, because the Germans need an answer. So suddenly, instead of at my level and other low levels, the Committee of Principals, that's those secretaries and their deputies, their immediate deputies, the Committee of Principals, had, now even they thought more about chemical weapons, but there still was the opportunity to get papers about biological weapons into that group. So this all happened long before the idea of a biological weapons convention came along, but it did stimulate a lot of thinking and interest. It at least justified guys who were interested in this anyway spending their good government time thinking about it. So there was a lot of accident and good luck along the way, but we got it. And then there was the question of toxins. The Joint Chiefs argued that toxins are chemicals. They're not living things. Botulinum is a protein. So they must be like chemical weapons, and they're not prohibited. The State Department argued toxins are made by germs. Not all toxins, but botulinum and many others are made by bugs. That's usually why you get sick, the toxins that are made by bacteria. So the State Department wanted to classify them as biological, which means they're prohibited, because we had the BW Convention, but did not yet have the Chemical Convention. So there's a big argument in the government. And Mel Laird, who was Secretary of Defense at the time, he chose the middle. We renounce toxins if they're made by bugs, but if the chemists can make them in a chemical factory, those we can use. So Henry and the president go off to Key Biscayne, which was the president's vacation spot. At that time, it wasn't like it is today, which is for tourism. It was an island owned by the Navy, a federal territory. So they take with them the position papers on this issue. I had written a paper called, you could go to my website and see it, or maybe my website is not on the web. I'm building up a big website. I think we haven't put it on the open web yet. But Janet can send it to you. It's called What Policy for Toxins? And in this paper, most of the papers, really almost all the papers I ever wrote for Henry, I tried to imagine I'm Richard Nixon. What arguments will appeal to me? Not the arguments I make because I'm me. I'm the arguments for him. And yet, I'm supposed to be a scientist. So I could say that an artillery projectile of 155 millimeter weighs 100 pounds. and the warhead with nerve gas in it or with a toxin in it is 4 pounds. So you still got to send all that metal. Using toxins instead of nerve gas in your chemical weapons doesn't make the weapon any lighter. It's true that the area of impact with a highly toxic toxin might cover a larger area, but still the fact that they have to be delivered by this great big piece of metal means that this ratio of toxicity is a misleading indicator. Second, none of the toxins work through the skin, whereas there are chemicals like VX that go right through the skin, other than just having to be inhaled. So for a toxin, there's none of them that work through the skin. You have to eat it or inhale it as an aerosol. But we already have a weapon, VX, that goes through the skin, much more effective. Also, and I have these other technical arguments, but then I read an editorial in the Washington Post by a guy named Steve Rosenfeld, who was on the editorial board. And it said, how can the president renounce typhoid? Because the Biological Weapons Convention was in existence. Only to embrace botulism. Well, I thought that's a pretty catchy line, but how can I get it into a paper that's supposed to be from a scientist? Because that's not a science statement. So I figured out, here's how I can do it. Each of the sections of my paper has a heading. What if I have the heading, the credibility and authority of the President of the United States? I didn't realize at the time, or maybe I did a little bit, but not fully, that a President of the United States looking at this little paper by this guy named Meselson, the only thing that's going to really catch his eye is my credibility and authority? Yes. What's he got to say? And he sees this line, how can the president renounce typhoid only to embrace botulism? So what they brought down with them from Washington was this paper in which Henry had written. Henry recommends option number 2, the Mill-Laird option, probably because he didn't want to get into a fight with Mill-Laird, because there are other bigger things he wanted from Mill-Laird. So sometime during that evening, when they're trying to decide this issue, which box would the president check? 3 boxes. They can't find my paper, and Henry wants to show it to the president. So they call me. I'm out at dinner with friends. So they called Paul Doty, asking Paul, because Paul and Henry were good friends, do you have Matt's paper? Paul had it, but he couldn't find it either. So at 11 o'clock at night, I'm still out. Henry calls Paul and says, the president has made up his mind. He's renouncing toxins, whether they're made by chemists or by bugs. What made up his mind was that thing in Matt's paper about the credibility of the president. So here are the arguments that, to me, were, the real arguments. And this other thing I stuck in only because they weren't scientific, but I thought it's a good thing to put in.
Yeah.
I should have realized, of course, what else would matter to a president? But that's, if I achieved anything for sure that was just pure me, it was that. Because Henry was recommending option 2. Nixon probably didn't give a damn. It was too, tiny little question. Yeah. But ask Janet for that paper. I think you'll like it. It's a very simple little paper.
So how effective do you think the bioweapons treaty has been throughout the years?
Well, there has been no biological warfare. There's been some terrorism. We had the Anthrax letters. Now, the Biological Weapons Convention actually is also directed against domestic terrorism because it obliges its states parties to take action within their own jurisdiction against the hostile use of biological weapons. And whoever was responsible for it, I think the case that was Bruce Ivins at Fort Detrick is a very strong case, and the arguments that it was someone else are very weak arguments, but I cannot prove who did it. I know a lot about it. I knew Bruce, not well, but I knew him. And a lot of nonsense has been said about this subject. My wife, Jean, wrote a book about it called American Anthrax, a very good book. On the other hand, you're asked to be how serious we should take it. Potentially very serious, but the anthrax letters was, 15 years ago? What was it? Was it 1990 roughly?
I think it was in the 90s, yeah.
It's more than 20 years.
Right.
It hasn't happened again.
Yeah.
If there was a huge threat, it would have happened again. Now, tomorrow morning we might read in the newspapers, it's happened again. What if 100 years? I mean, history has got to be good for something. 100 years from now, it still hasn't happened. 200, 1,000? I mean, you've got to apply some degree of rationality to these issues. So the first point I would make is, it can't be that big a threat, because what that episode did is it shows it's easy to do. You just put the spores in envelopes and mail them. It works. It killed a lot of people. So a copycat, if somebody really wanted to do it, had basic training, reads the literature, you could do it. And nobody has. So that's got to mean something. It means two things. It means that the present threat, at least up till now, up to this moment, obviously has not been great because it hasn't happened. But the second thing it says is that motivation is key. I say that because if there was someone out there with at least reasonable amount of training in bacteriology who wanted to do it, it would have happened. So if I could generate 10 guys with that kind of training who want to do it, it would have happened. So the question of intent is what a chemist would call rate limiting. And so that's what you want to protect. You want to keep people from just wanting to do it. Now, one way we do that is by training hundreds of thousands, even millions of people, mostly men, to use explosives. And then we make it easy for them to get attack rifles. So if you want to do a lot of rampaging, you've already been trained to use weapons if you've been in the military. They're easy to get. It doesn't take any time, because if you suddenly decide you want to kill a lot of people, if you were told, well, you can do it if you still want to do it after a month. After a month, you might have decided, I really don't want to kill a lot of people. But here's something you can do on the spot. When the mood comes over you, you can do it. Whereas with BW, if you hadn't already planned on it for a long time, learned how to do it, go in development, you have to have the intention to do it for a long period of time. In other words, there are all kinds of arguments why motivation is the key here, not availability. It's easy to get a hold of anthrax. It's in the soil. I could select for it. It's easy to grow it. It's easy to get the spores. It's easy to dry them. So that's the key, is intent. So that means that if we yell and shout in the newspapers, which we don't, thank goodness, but if we were to do that, there's a biological threat, it's terrible, this will put the idea into someone's head. So my view has always been, don't shout about it. Do whatever you do it quietly. None of this shouting is going to do anything except maybe give someone the idea to do it. So I don't think the threat is very great compared to other threats we have. The most subtle threats are the things you don't realize. Over long, long periods of time, what is going to happen to the human genome? I mean, basically, that's the only thing we got. I mean,
right.
that's one thing.
us.
Without that,
right. So looking more towards the future, I mean, there's new technology coming out in lots of different places. I know you mentioned Franklin Stahl worked in phages, and now phages can be used for phage therapy. There's also the computation revolution. What are some of the things that you think are most exciting going into the future? What are some of the things that you think are most concerning?
By phage therapy, you mean just because phages kill bacteria?
Yes.
I don't think it's very good.
No? Why?
Well, first of all, because there are many compartments of the body where phages cannot exist. So there's some kinds of bacterial infections which are in places where you can put phages, but there are other places in the body where you can have bacterial infections. You can't get phages in there, or if you did, they wouldn't be active, they wouldn't work. Second, it sort of limits you to the gut. So the only real motivation for wanting to think about them is that people are becoming resistant to a lot of antibiotics.
Right.
That's the one way in which you might, at least in my opinion, I haven't thought a lot about this, but that's the danger of talking to guys like you. You tend to say things that you're not sure are right. But off the cuff, I would say that there is certainly a problem of resistance to antibiotics. That's for sure. And in a way, using phages as therapy is related to the larger question of the human biome generally. Because if you start using phages as antibiotics, and we have a lot of bacteria in us which are either without much effect on our health or which contribute in a positive way to our health, we need to be sure that the phage therapy doesn't muck that up. So those phages must not go after any of the bacteria in our biome, any of the accessible bacteria in our biome. And what's left? Salmonella poisoning. Cleaning up the food chain. I mean, what is this? There's a recent recall of what? I forget what it is.
Romaine lettuce gets recalled.
Wait, is it lettuce?
A lot of the time, I've heard a couple of recalls about romaine lettuce, yeah. And there's also been multiple meat recalls and things like that for similar concerns.
Now, society has only a certain amount of capital in various areas, research, border patrol, whatever you want to call it, so that effort spent on inspecting the cabbages, lettuces, or spent on phage therapy are not quite fungible with respect to each other. So you're right to ask about the viral therapy on its own. I could be wrong, but I don't see the... Yeah, I guess the best thing is to say I could be wrong, but I don't see that much potential. It's been tried, for many years, off and on. In any systematic way that would really find its true potential, I guess not.
Yeah, it was my understanding that it started to go through a little bit of a renaissance recently, but I'm not an expert on it.
Are gastrointestinal infections a big problem?
I don't know if it was used. I've heard it used with burn victims a lot of the time, like on the skin.
Burns?
Yeah.
Well, why would that be?
I don't know the details.
I don't know anything at all about that.
Yeah.
The main thing in burns, I would think, well, you don't want to get them infected.
Right, and I know that the main thing was
there's a lot of dead tissue, and you'd like something to come along and eat that up. But phages don't eat, even though phago means I eat.
Right. But I think it was to attack the infections in the burn sites.
Yeah, burns do get infected.
Right. And I also know, not exactly with phage therapy, but similarly using other viral vectors to do things like gene therapy and things like that, which have also been happening more and more often. So I guess it's kind of a last question. You've worked with so many amazing people and are an amazing person yourself. How does it feel to have done all of these things, to work with Henry Kissenger, with Richard Feynman, with Freeman Dyson, all of these amazing people that have been around you, and all of the amazing work that you have led? are you happy with your life? Are you happy with how things have gone? Or do you just feel like there's more work to do?
I should be. You see, I had a most marvelous wife who died. Incredible person. So I keep very busy because I don't want to just fall into a well of despair. Yeah, and when I have some kind of project, whether it is to get something done in the world of human activities or just in the lab, it's sort of like you're floating free. It's hard to explain. You're in a field-free space, I guess. Nothing's tugging on you. I don't know how to explain it. I'm very fortunate because even as a kid, my parents, I had the terrific laboratory in what had been the garage, but you couldn't get a car in there anymore because I filled it with lab benches and shelves and stuff. My parents, neither of them went to college, but they had they trusted me. So I was very fortunate in that respect.
Yeah. And you've gotten to bring those workbenches all around the world now, I guess, and kind of with all the work that you've been doing.
But I would like to get the realization that we need to filter the air really well established. I care a lot about that.
Right.
If I could really show a way to cure solid tumors, that would be wonderful. Aging, I don't know. If people could live till age 300, is that so good?
Yeah, a lot of people argue about that. I personally would love to live to 300, but a lot of people don't agree with me.
Has anyone ever seriously tried to think out a society, what would it be like, where longevity was to say 300 years? We can't do that right now over this table, but in principle, you ought to be able to think hard of what the implications would be.
I think a lot of people argue back and forth about whether people would want to continue working past retirement age. Would they still feel motivated to continue to do work like you do, or would a lot of people say, "I've done my time"?
The first implication is there'd be a lot more people.
Right, of course.
So that means that the resources upon which we depend would have to be greatly increased, or else a large fraction of the population would have to live in horrible circumstances, not enough food, etc. So the first things I think could be dealt with by economists. So if you had a summer session of a bunch of really smart economists, they could probably tell you what would happen to just the flow of goods, of demand and supply. And it would be a very big effect. After you got that, I think you want to start with that, because those things would all feed into the other questions. The impact of great longevity on the availability of things all the way from food and air and water to fancy things like Goya prints and fancy cell phones, would all be impacted in ways that can be at least roughly quantified by economists. So I think you want them to do the first work and only then bring in other kinds of people to think about other things. Sociologists, certainly, what happens to family structure? You know, your great-great-grandchildren. Who gets invited to your parties? Yeah, I think you would think it out and have a pretty good idea. And maybe the greatest impacts that people would really care about initially would be these economic ones. Like if the amount of energy production now from all sources is whatever it is, and you quadruple the population of the planet, or would fertility go down? Do people still want to procreate when they're 120 years old?
That's a good question. I
would
not know.
I mean, the 120-year-old people we know now don't do a lot of procreation. I think if you could have greater longevity, but not greater fertility. Because to increase the population of this planet by a factor of 5, you really don't want to do that.
And then you run into the question of if you have this increased fertility, do you now have...
If women lose their fertility just as they do now, by about age 40, the prospect of extending life a great deal does not automatically reach the conclusion of huge increase in population.
But you would have a subtle increase in population because you wouldn't have a death rate anymore.
You'd still have a big increase.
Right.
But nothing compared to 300 years of reproduction.
Yeah.
Because then the number of individuals produced by one individual or by one couple, instead of being 2 or 3, unless you had legislation that somehow you have to sterilize people after they've had their second child.
Do you think there would be...
first child.
You think there would be families that would want to have multiple generations of children? I mean, imagine having children 50 years apart.
Not voluntary. It's not... I mean, it just happens.
I guess that's true. Yeah.
How many children are actually planned?
No. That's an interesting idea.
I think we're getting beyond my expertise. I don't know about you.
Okay. Well, I appreciate so much that you came on today. I really enjoyed talking with you. I think this was great.
I have one more thing I want to talk about.
Sure.
Which is we need people, talented people, who are thinking about the more distant future. Elon Musk apparently is. So, for example, I have a friend who advises undergraduates at Harvard. Quite a few graduates of Harvard in later life will have positions of influence in the world of politics or the world of business or in the intellectual world. I think that we should do our best to try to encourage the more promising students. I know this sounds a little bit elitist, but you have to admit that there's differences between students. To think hard about long-range questions. That maybe there should even be a course for undergraduates. You could lure them into it by some fancy name, like existential threats. And since everybody has to get some general education credits, you get that into the GE program. And now the first half of the course could be taught by geologists and physicists and cosmologists. An existential threat is if a great big chunk of metal from outer space hits this planet, wipes us out just like the dinosaurs. So that means we need to make sure that NASA is looking carefully to see anything that looks like it might be on a path to hitting us. And if it is, let's figure out how to deflect it early, because you need much less work to deflect the trajectory if it's far away than if it's right up close. So that's one kind of existential threat. And then there's pandemics and other existential global climate change. But then there are other kinds of, and we should try to encourage students to be interested in these long-term problems by offering courses. Because very often what you get interested in for later life is something that happened to you when you're an undergraduate. So I think there should be courses that would cause some of the most talented and imaginative and effective students to include this in the interest they will have in their later life, the future of our species. And you can do that by offering courses for which they get credit. In fact, you could require the course. That would be too brute force, because the students who'd be attracted to take that course probably constitute a large fraction of those who would ever want to do anything about it in the first place. So you don't need to require it. But it seems to me that it should be a prominent offered course. The basic idea being, amongst these graduates from places like MIT and Harvard and Princeton and Yale, Stanford, are going to be people with influence. Do we want them to have that influence over the design of the Coca-Cola bottle? Or do we want at least some of them to be thinking about the long-term future of our species? Well, clearly, we'd like to funnel as many of them as possible. I don't see where there's any limit. Well, maybe there's some limit, but it seems to me that if we have twice as many such people than we really have now, this is by no means too many. So that's the last thing I wanted to say. I think that the major places where bright students are likely to end up, like MIT and Harvard and Yale and Princeton and Stanford, there should be a course on existential threats, including not just things that are physical threats, but other kinds of threats, threats to our humanity, especially. We've got to worry about the future.